Scientific progress often happens by reducing a vast range of complex phenomena to shared rules. When we find the underlying principles that connect separate events, the world becomes more predictable. Every living system is constantly flooded with environmental data. To survive, an organism must navigate a chaotic stream of light, sound, and touch, responding to what matters while ignoring the rest. This capacity to find what matters is called relevance realization. It is the core requirement for intelligence, the prerequisite for every perception, prediction, and action. Traditional cognitive models treat this filtering as a specialized task. They assume a dedicated central controller sorts the data and decides what's important. But neuroscience has entirely failed to locate a physical equivalent. There is no specialized relevance module inside the human brain. To solve this mystery, we have to look past high-level cognitive models and examine the universally accepted, low-level mechanics of the nervous system. This creates a central paradox. If the brain lacks a dedicated organ for determining relevance, then intelligence likely arises from the self-organizing dynamics of its standard biological parts. Let's start with the uncontroversial facts of neuroanatomy. At the microscopic scale, the nervous system is a web of cells communicating through electrochemical signals. Neurons integrate excitatory and inhibitory signals. Crossing a threshold fires an action potential. Below this line, massive subthreshold populations remain silent but highly active, holding a charge that influences future behavior and reflects the system's current state. This electrical landscape is never static. Incoming sensory data continuously nudges these resting potentials up or down based on what the organism sees and hears. Internal variables like memory and physical baseline states also feed into this calculation, summing together within the membrane voltage of the cell. This distinguishes biological systems from standard digital computers. While a computer relies on binary switches, a brain relies on the continuous integration of these shifting potentials. Biological neural computation is defined by this dynamic landscape. The activity of the brain is as much about the potentials hovering below the threshold as it is about the spikes crossing it. We can scale this view up from the single cell to the larger architectures of the brain. Cells organize into regional networks with specific shapes. Neural topologies organize information into physical maps mirroring input structures, like retinal layouts or sound frequencies. These are essential, preserving functional relationships and ensuring sensory data stays organized as it moves through the system. These maps change through Hebbian plasticity. Neural connections strengthen or weaken based on firing together. When activation patterns repeat, physical connections thicken, making that specific pattern more likely to activate in the future. Experience leaves a measurable physical trace within the tissue. Learning is the ongoing reorganization of these biological circuits. This turns neural networks into dynamic maps that mold their own physical structures to mirror the statistical regularities of the outside world. These learned topologies do not operate in isolation. They are driven by competitive excitation and inhibition, where different neural populations effectively fight for dominance. Many different neural representations can exist at once, but only a winning subset accumulates enough excitation to cross the firing threshold and drive motor behavior. This competition is also shaped by embodiment. Cognition is inseparable from the physical body it inhabits. Internal variables like hunger, hormones, and fatigue act as constant signals that bias neural competition, changing which populations are most likely to reach the activation threshold. This is our biological toolkit: subthreshold integration, mapped topologies, structural plasticity, competition, and bodily feedback. Each of these mechanisms is a staple of modern neuroscience. The next step is to ask what happens when they all operate simultaneously in a single unified system. We are now moving from established neuroscientific fact to the proposed theoretical framework of tokenization theory. This framework does not introduce any new biological mechanisms. Instead, it explores the consequences of combining the mechanisms we just described. The central construct of this theory is the token. In this theory, a token is a specific, learned topological organization of neural activity. It's the physical structure encoding a behavioral regularity through past experience. Multiple tokens exist simultaneously in the subthreshold landscape, sitting in a partially activated state, waiting to be recruited. This suggests the brain is packed with latent structural representations, constantly hovering near the edge of activation and ready to be deployed by the next incoming signal. This brings us back to the original paradox. Without a central executive, how does the brain know which token is relevant to the current moment? Relevance is dictated by competitive excitation. When sensory and bodily inputs converge, the most energized token breaches the threshold, suppressing surrounding clusters. Once it breaches this threshold, it assumes control of perception and behavior, becoming the dominant influence on the organism's next action. Relevance realization emerges from this threshold competition. The dynamics of the network itself perform the sorting, without the need for a specialized selection mechanism. This framework implies that attention, prediction, and even symbolic reasoning are not separate faculties, but manifestations of this same underlying excitation dynamic. The established neurobiology does not prove tokenization theory, but it provides the rigorous grounding needed to test it. It offers a way to see how simple biological primitives might naturally self-organize into intelligence.